Hi everyone, uh, welcome and thank you for coming to the second Michael Lamb lecture. We have approximately 250 to 300 people in attendance, and the event is being organised by myself on behalf of the Psychology Department and the Psychology Society, led by Erica Dukainen, who currently can't be here at the moment, but we're hoping we can bring her into the lecture in due course. My name is Chris Watkins. I'm the Head of Department of Psychology and Forensic Science, and it's a great pleasure to introduce our speaker for the second Michael Lamb lecture as part of our annual lecture series, named after Abertay's honorary graduate, Michael Lamb, currently Emeritus Professor at the University of Cambridge. So Dr. Serena Scarpelli is a psychologist and cognitive behavioural psychotherapist, and currently a postdoctoral researcher at the Department of Psychology at the University of Rome Sapienza. Serena is a member of the Laboratory of Sleep Psychophysiology, directed by Professor Luigi De Gennaro. Serena gained her PhD in 2017. And she is interested in her research in what happens to the brain when we dream, and has examined this in clinical samples, such as in people with Parkinson's disease, narcolepsy, Alzheimer's, and among people addicted to smoking. Serena works on the International COVID-19 Sleep Study and has published over 50 peer-reviewed papers in scientific journals. Her talk is titled "What About Dreams? Advances in Our Understanding of the Neurobiological Mechanisms of Mental Sleep Activity." So Serena, it's great to have you here. Welcome to Abertay, and thank you for continuing the tradition of our annual lecture series. I will hand over to you and your slides just now.
Well, thank you, Christopher, for your presentation, and thanks for this kind invitation. It's a great pleasure for me to be here, and today I'm talk about about the fascinating world of dreaming. And well, I'm going to talk you about the birth of the scientific study of dreaming, and then we will move on more technical aspects of dreaming, and I'll talk you about the approaches that scientists use to study dreaming, from neuropsychological to neuroimaging studies and electrophysiological approach. Well, first of all, let me start by giving you some background information about the birth of the scientific study of dreaming. Well, dreaming is a particular kind of mental activity occurring during sleep. It can happen many times per night, and it is characterized by different degrees of emotional intensity, bitterness, visual vividness, and also by different level of narrative complexity. In particular, we could have both dreaming with a complex narrative structure, but also dreaming composed by more fragmentary thoughts. And from the discovery of REM sleep, many investigators believed that dreaming was just an epiphenomenon of REM sleep. Indeed, many authors, many investigators, when asked people, have you had a dream after a REM sleep awakenings, found that in more than 80% of cases, subjects had a dream report. But this correspondence has been refuted by thoughts who changed the way we ask for dreaming to the subject. Specifically, this author changed the question from "Have you had a dream?" to "What was passing through your mind just before you awake?" And well, in this way, also after an awakening from REM sleep, many studies found that more in the more of the 70% percentage of cases, we could have a dream report after no REM sleep awakenings. So dreaming is not just an epiphenomenon of REM sleep, and the neuropsychological studies confirm this. In particular, REM sleep and dream recall are modulated by two different systems, two different brain mechanisms. Patients affected by brainstem lesions in the regions related to REM sleep control, to REM sleep generation, still preserve their dreaming. And in parallel, dream recall totally disappear in patients with focal forebrain lesions and without any impairment of REM sleep. So these first neuropsychological studies highlighted a double dissociation between REM and dreaming, confirmed that dream recall is possible both after REM and no REM sleep. So the subsequent studies focused on the qualitative dream features from REM sleep and non-REM sleep awakenings. And many authors emphasize that dreamlike oniric activity is characterized by more vivid and visual components, more bizarre, more length, and also by higher emotional intensity. On the other hand, like no-REM oniric activity appears to be characterized by more fragmentary thoughts and realistic contents and lower emotional resonance. However, also this dichotomy was questioned, since a significant portion of dreams reported from no-REM sleep stages shows several features that are peculiar of dreamlike reports, dreamlike meditation. And also some studies that taken under control the total word count, the length of dream reports, evidenced that the control of word count makes similar REM and no REM reports. Well, now let me to ask you, can we directly access to dream experience without interfering with dreaming itself? Well, the answer is no, we cannot. We cannot have a direct access to dreaming. And for this, we could introduce the paradox of the Schrödinger's cat, because when the cat is in the box, we cannot know if the cat is alive or dead when it is in the box. But to have some information about the cat condition, we have opened the box, and so we change in this way the cat condition. Well, for dreaming, it's quite the same thing, since when the subject is producing a dream experience, the subject is still asleep. And to have some information about the dream contents, we have to wake up the subjects. And so the real object of study is not dream per se, is not dream in the moment in which it's producing, it's generating, but it's the dream recall after awakening. So we must not forget these three points. Dreaming is not directly observable. Researchers are able to obtain information about the dream activity just requiring a dream report to the individual when he is awake. And finally, detecting the exact moment in which the dreams are produced during sleep is very difficult. Well, Let's move on to the neuropsychological and neuroimaging studies. The first neuropsychological studies on dreaming aim at evaluating the correlations between dream features in brain damaged patients and anatomical lesion sites. Specifically, Solms in 1997 reviewed more than 100 cases of dream cessation, of dream loss, showing that two systems are involved in dream activity. The first is the parieto-temporal-occipital area, and second is the ventromedial prefrontal cortex. In particular, the temporoparietal-occipital junction is responsible for the visual components of our dream scenario, of our dream activity. But these studies are not free from flaws, since in this study, dream recall was retrospectively collected, and this would imply some memory biases. Also, dream recall in patients could be affected by pharmacological treatments, since medications in these patients, for instance, medications after a stroke, could have some impact on the cognitive processes, and so also on mental sleep activity, also on dream production. And finally, we have no information about dream recall during premorbid conditions. So we cannot know if we cannot know how the subjects dream before the stroke, before the lesions. Well, then neuroimaging studies were aimed to evaluate if dream contents reflect the degree of brain activation during REM sleep. And most of these imaging studies show that REM sleep is characterized by a general high cerebral activation, in particular in correspondence of thalamus, hippocampus, amygdala, occipitotemporal area, and orbitofrontal cortex, as compared with the slow wave sleep. And also, some authors highlighted that specific dream features are related with the specific activation of some areas, some regions. For instance, predominant visual components is related with the higher temporal-occipital area, where higher activation in the temporal-occipital area, and also the high emotional intensity and specifically fear appears to be related with the higher activation in the limbic system, so in the amygdala. And also the lack of insight, the time of distortion that could have in our dream contents, and the amnesia of our dream contents after awakenings could be related with the lower activation over the prefrontal cortex. But also, these studies are not free from limitations, since they were still bounded to the implicit assumption that sleep meditation strictly depends on REM sleep physiology. So these studies remain bound to the older REM sleep dreaming equation. And also, these studies add a bias of timing, namely a precise definition of the time coupling between sleep stages and the actual occurrence of dream experience is difficult, and we can have this information. We cannot have this information. So, more recently, scientists are focusing on trait-like factors related to the dream recall. Namely, they aim to find whether specific stable characteristics of the neurobiological mechanisms could be related with some characteristics of our dream contents, of our dream reports. So along this vein, the Gennaro colleagues recently found that some structural features of the amygdala and the hippocampus are related to qualitative dream characteristics among people that frequently recall their dreams. And the volumetric characteristics of hippocampus and amygdala appear to be related with the emotional load, the visual vividness and bitterness and total word count of the dream report. We have to remind that amygdala and hippocampus are also linked to mechanisms that are responsible for emotional memory processes during wakefulness. So there is a sort of continuity between our mental processes in different states of consciousness. And with a slightly different approach, Michael Lamb colleagues recently studied differences in the activation, the cortical activation between high recallers and low recallers. And they found that higher activation of there is a higher activation of temporoparietal junction in high recallers, and this region is also part of the default mode network that during the wakefulness, during our waking state, is responsible for daydreaming and mind wandering. So again, we have a sort of continuity between the neural mechanisms based on our cognitive processes between wakefulness and sleep. But the best method to study the neural basis, the neural correlates of dreaming, dream recall, is the electrophysiological approach. Specifically, the polysomnography with provoked awakenings in laboratory is the gold standard method to study neural basis of dream recall. Well, here we have an example of a standard protocol to study the electrophysiological correlates of dream recall. The subjects in laboratory undergo a PSG montage with cortical electrodes, and here we can have a standard montage, but also in recent period we have also a high density, so a lot of electrodes, a lot of derivations that allow researchers to have a high spatial resolution. And polysomnography also include electroculogram to study the eye movements, and also electromyogram to detect the changes in the muscular tone. And well, the subjects could have an undisturbed night, but also a nap recording or multiple awakenings during a night. And after each awakening, we could have a recall condition, so we can obtain a dream report, but also we can have a no recall condition. Well, one of the most representative study in this field is the study by Marzano and colleagues realized in our lab 10 years ago. These studies was aimed to identify the hazy sleep pattern predicting dream recall in the last 5 minutes of sleep before awakening. And here, the authors found that during the last 5 minutes of no-REM sleep, before the awakening, we have a lower temporoparietal alpha activity. And during REM sleep was found a higher frontal theta activity. So you have to know that the same pattern, the same AG patterns were found during wakefulness in association to good performances in declarative and episodic memory task. So if we consider it, dream recall as a peculiar kind of episodic memory, as an episodic memory traces, we could say that these results provide support to the continuity hypothesis in its neurobiological perspective. In other words, wakefulness and sleep share the same neural mechanisms from the encoding, the elaboration of episodic memories. We have the same patterns to recall a pieces of our memories and memory traces during sleep and during wakefulness. But this study has a main problem, main limitation. It was carried out by a between subject design. So the recall condition and no recall condition have been obtained from different subjects. So we can take under control in this study the trait-like factor. And so by using a between subjects design, we cannot understand whether the EEG correlates of dream recall are related to state or trait-like individual differences. Stage-like differences, namely the physiological scenario related to the specific sleep stage of awakening, or trait-like factors, namely the stable characteristics of the subjects that are participants in our experimental session. So to disentangle this issue, we more recently carry out within subject study with REM sleep awakenings. Namely, in this study, we obtained the same from the same subjects, both recall and no recall conditions. So in this way, we can take under control the trait-like characteristics. And we found again that the frontal theta activity during REM sleep is related to the subsequent presence of dream recall. And again, these results are in line with the continuity hypothesis between sleep and waking and wakefulness. And also, this within subjects protocol allow us to state that EEG patterns of dream recall depend on state-like factors. Well, but when we repeat the same protocol with no REM sleep awakenings, we did not find the effect on the alpha power as in the study by Marzano and colleagues. But we found by a within subjects protocol that we have differences over the delta activity, specifically lower frontotempoparietal delta activity. So as low oscillation are associated to dream recall. So a greater level of arousal, of cortical arousal, is related with the dream recall. This is consistent with the so-called activation hypothesis that states that dream recall will be facilitated by a greater level of arousal during sleep, represented at an electrophysiological level by greater AG high-frequency activity. Something like this was posited by Kulak and Goodenough in the 1976 with their arousal retrieval model that posited that the mental activity during sleep could be memorized only when sleep becomes more fragmented and short awakenings occur during sleep, allowing the dream material to be elaborated and transferred from short-term memory stage to a long-term memory storage. And so more awakenings and more activated brain promote the subsequent dream recall. Also, this is consistent with a study by De Gennaro and colleagues that reported that a night of recovery sleep after a total deprivation that was usually characterized by reducing awakenings, but also by a greater level of slow oscillation during non-REM sleep, almost totally abolished dream recall after the final morning awakening. So in the complementarily, we found in this study that when the sleep is characterized by a greater level of delta activity and by reduced intra-sleep wakefulness, the dream recall was almost totally abolished after awakening. And this was also confirmed, the activation hypothesis was also confirmed by this recent study published on the Nature and Science of Sleep, in Nature and Science of Sleep. Since here we found with multiple awakenings during the night, REM sleep awakenings, that higher occipital beta activity in the posterior area was associated to dream recall. So beta activity is a very rapid frequency and represents a higher level of cortical activation. So we can say that the occipital area and in particular the posterior area could represent a knot zone responsible for dream experience, and this is also in line with the results by Solms. This idea of activation in relationship to dream recall was also confirmed by a study published in Nature Neuroscience from the group of Tononi. And in these studies, they found that in both non-REM and REM sleep, reports of dream experience were associated with local decreases in local frequency activity in posterior cortical area, both in non-REM and REM sleep. And in parallel, higher frontal temporal gamma activity in REM and posterior rapid gamma activity in non-REM sleep were related with dream experience. Also, similar patterns were found in narcoleptic patients that are a producer of dreaming. They produce a lot of dreams. And also in this study, we found that cortical activation during REM sleep and non-REM sleep predicts dream experience. Well, then we wonder whether this EEG correlates, remains stable also in older adults. So we have studied also the relationship between dreaming and aging. Actually, many findings disclose an age-related reduction in dream recall frequency. And some authors reported a relationship between the decline in dream recall rate and general cognitive impairment during aging. Even if other studies have found that this reduction starts at 35 years, so among young people. Another hypothesis is that this age-related reduction could be ascribed to the method used in these studies. Indeed, these studies were carried out by retrospective questionnaires, so it could be some memory biases. And also, additionally, another hypothesis is that during aging, it could be a reduction in dream salience, namely, older adults could have lower interest in their dreams, and this could explain the drop in dream recall frequency. It's very interesting the fact that a later decline in dream recall frequency was observed for women compared to men. So women, females appear to be more interested in their dreams across the lifespan. Concerning the age correlates of dream recall in older adults, we carry out this study in order to understand if the same age patterns that we identified in younger remain stable also in older adults. And again, we found that higher frontal theta activity during REM sleep predict dream recall both in younger and older adults. So again, we have a result in line with the neurobiological continuity hypothesis, because this same pattern was identified in during wakefulness in relationship to good performances in declarative and episodic memory tasks. And also during non-REM sleep, we found again that lower delta activity during REM sleep predicts dream recall in older adults. So again, during REM sleep, the greater activation in frontotemporal and in the posterior areas predicts dream recall in older adults, as well as in young adults. And furthermore, in this study, we also correlate the level of arousal with the qualitative aspects of dream report in older adults. And we found that the higher level of arousal is positively correlated to vividness at dream length. And also some characteristics of the sleep macrostructure, of sleep architecture, appear to be related with the dream features. Specifically, intra-sleep wakefulness is related to more vivid dream contents, and also lower sleep efficiency, so namely poor sleep, is related to dream length. But today, I cannot leave you without telling you about the relationship between dreaming and emotional processing. Well, in your daily life, you have certainly experienced it that some events can be incorporated in your dreams, in your dream scenario. And this is consistent with the continuity hypothesis in its psychological perspective. The continuity hypothesis proposed that personal concerns and conceptions characterize the waking thoughts have their own continuity in sleep. And here we could have Also the incorporation of older memories, dream lag effects, but also the incorporation of very recent waking life experiences. Namely, we can have a day residue. But the most important thing is that the emotionally relevant waking experiences are more easily incorporated in our dreams. Michael Lamb and colleagues have recently tried to find the AG correlates of this incorporation of this phenomenon, and they found that the number of references to recent waking life experiences in REM dreams was positively correlated with frontal theta activity in REM sleep period. And also, the emotional intensity of recent waking life experiences incorporated into dreams was higher than the emotional intensity of experiences that were not incorporated. So the emotional resonance is very important. But what about nightmares? Nightmares and bad dreams can occur in comorbidity with psychopathological condition and sleep disorders. Actually, there are very few studies about the EEG, the neural correlates of the nightmares. The available literature reported that higher autonomic and electrophysiological activation may provide the physiological background to the nightmare occurrence. And this is consistent with the self-reported experience of the greater emotional and physical activations during the nightmare production. On the other hand, also, the nightmares could be related with, could be a symptom in the post-traumatic stress disorder. This is an anxiety disorder caused by stressful or other adverse events, and is characterized by hyperreactivity to emotional stimuli and an inability to extinguish traumatic memories. Many authors highlighted that sleep disturbances and recurrent nightmares are recognized as the hallmark of PTSD. And in this view, recurring nightmares could represent the failure in the processing of traumatic emotional memories. But on the other side, we could have another phenomenon, the lucid dreaming. Lucid dreaming is the experience of being aware of dreaming during sleep, and it may be considered as a sort of coping strategy to face with the negative emotional contents of dream experiences. So it could have some implication for emotional regulation. Indeed, greater lucid dream frequency in narcoleptic subjects promotes relief during their nightmares. And also, some studies suggested that lucid dreaming could be useful as an intervention for nightmare disorder. The idea is that the high degree of awareness of own dreams increases the possibility to regulate, to modulate our emotional contents. And for instance, during the pandemic, some studies found that greater lucid dream frequency was found in narcoleptic and healthy subjects, in particular during lockdown. Well, overall, we can say that dream activity can be considered as an expression of mental health, of our mental health, of our well-being. Indeed, in our studies on pandemic dreams and pandemic nightmares, we found that there are greater nightmare frequency among people during the pandemic, and in particular, when we investigated the nightmares in the COVID-19 patients, we found that COVID-19 patients had a higher nightmare frequency than controls, than people without COVID-19. Well, we are near to the conclusion, but before concluding, I think that it could be important to introduce you one of the main perspective for the future of the dream research. Namely the Parasomnia-like events that could represent a viable access to dreaming. Well, the dream enacting behaviors are any acting out of a dream during sleep, characterized by motor, emotional, or verbal components, and they may be considered a direct observation of dream experience while the subject is still asleep. The study of parasomnias or parasomnia-like events such as REM behavior disorder, sleepwalking, nightmare disorders, or sleep talking may provide new insights about dreaming. In particular, in this study published in Journal of Sleep Research by Rivera Garcia and colleagues, the authors, in a pioneering way, study for the first time a sort of parasomnia-like event in healthy subjects. And he focused on emotional facial expression during REM sleep. And the authors found that the activation of corrugator and zygomatic muscles are highly associated with dreams characterized by negative affect. Also, some studies by Arnold and colleagues highlighted that parasomnia and parasomnia-like events could have some role in memory processing during sleep. Well, in these studies, sleepwalkers are able to replay the recently trained behavior during the parasomnia episode. Look at this video together. Here we can see the protocol of these studies, and we will see three parts. In the first part, there is the execution of the structural motor sequence by a waking control. In the second part, we will see the repetition. The subjects was required to repeat the motor sequence just before the good night. And in the third part, we will see a sleepwalker that during no REM sleep, namely the patient is still asleep, we will see that the patient, the sleepwalker, repeat some pieces of the motor sequence. So look at this together. Here the subject is still asleep. This is a sleepwalker. Well, this study, the results from the studies supported the idea that dream enactment may have a crucial role in memory processing during sleep. And well, we think that this is a very promising way to have some direct access to our dream contents. Well, I think that today I gave you too much information, so let me sum up the main points. First, dream is a composite phenomenon with different degrees of emotional intensity, thoughts, bizarre and vivid elements. And most importantly, dream can occur in all sleep stages. Then, we cannot directly access to dream per se. The real object of many studies, of almost all studies, is dream recall. Dream could be considered as a peculiar form of episodic memory. Different states of consciousness share the same neural mechanisms from the elaboration of episodic memories, and this is consistent with the neurobiological continuity hypothesis. Also, dream recall is promoted by high level of cortical activation and sleep fragmentation, and this is in line with the so-called activation hypothesis. And moreover, young and older adults share similar EEG correlates of dream experience. Also, our dreams contain some waking life experiences, especially concerns and emotionally relevant events. And this is consistent with the continuity hypothesis in its psychological view. Recurring nightmares could represent a failure in the processing traumatic emotional memories. But on the other hand, the high degree of awareness of own dream, as in the case of lucid dreams, increases the possibility to regulate the emotional contents in our dreams. And finally, the parasomnia-like events could be interpreted as dream enacting behaviors, representing a promising approach to directly access to dream production. Well, this is the take-home messages, and I think that it's all for me. That's all for me, and I'm happy to answer your question. I want to thank you for your attention, and I want to thank my collaborators and Prof. Luigi De Gennaro, and that's all. Thank you.
Thank you, Chris. Hello, everyone. I'm Erika, the current president of Abertay Student-Led Psychology Society, and I just wanted to say a quick hello and thank you after finally being able to join after some technological issues. Anyway, thank you, Chris and the psychology department for collaborating with our society to carry out this talk. And big thank you, Serena, for such an interesting talk. I really enjoyed it. Anyway, that's all from me. Thank you for everyone for coming here. It was great to see such a big interest.
Hi, thank you very much. So I'm delighted to give the vote of thanks for this lecture. Dreams isn't my personal area of research, but my goodness me, what an absolutely fascinating lecture that was. I was absolutely riveted throughout, and thank you for your questions, some absolutely great questions. It was particularly interesting to me. One of the things that I always thought dreams only occurred in REM sleep, so it's interesting to know that they occur throughout sleep, and all the time I was also thinking the differences between my husband and myself. He has very vivid recall of dreams and can give me chapter and verse, whereas for me they fade incredibly quickly. So it's interesting to see how our brains are so different. So thank you very much indeed, Serena, for an absolutely fascinating talk. And I'd just like to add my thanks to the organisers as well, the student-led Psychology Society and the staff of the Division of Psychology and Forensic Science, and of course to Chris for chairing this today. I think we've learned an awful lot during the past 2 years, and it's great to see such an event working so well over video conference. Erica's problems aside at the start, but hopefully everybody's been able to participate and listen. So I think we're just going to have to hear your virtual clapping in the background, because it's not possible to give you a possible physical clap. But I guess I'll just do that from here on behalf of everybody. So thank you very much, Serena, and appreciate your time.